So now we will discuss about gram stain. What is gram stain? What is the use of gram stain in microbiology, and how does it help us to understand or study bacteria in microbiology? Let's see. Gram staining is basically a staining technique that is used to study the morphology of bacteria. Morphology means how the bacteria looks like under a microscope. So it helps us to differentiate the bacteria into two major groups. That is, either they are gram positive or they are gram negative. When we call them as gram positive, if they will stain purple using a gram stain, we call them as gram positive. In case they appear pink under the microscope after gram staining, we call them as gram negative. Why this differentiation is important? This differentiation is very important because it will help us to select the right kind of antibiotic for treatment if we are recovering these organisms from the patient. There are different type of antibiotics like linezolid, vancomycin, penicillin that will work on gram positive organisms, but it will not work in case of gram negative. Whereas there are other antibiotics like meropenem, colistin, which will work on gram negative bacteria, but it will not work on gram positive organism. So what is the procedure for gram stain? For making gram stain, for doing gram stain, you have to first prepare a smear. A smear means that you have to apply the colonies or bacteria on a glass slide. Then only you will be able to stain it. So how do we make a smear? We take a glass slide, mark the area where you are going to make the smear, label it with the name or ID, whichever patients or samples organisms you are going to take, and then start making the smear. For making the smear, you will need 1 to 2 drops of normal saline. Put it on the glass slide and then take the colony. The colonies or the bacterial colonies, they are shown on the left side on the blood agar plate. You use an inoculating loop. Inoculating loop is shown in the center. You use this loop, pick up the colony and then immerse or emulsify it in the normal saline on the slide. If you are using a direct sample, then you can just take the sample like sputum, pus, whatever sample it is, and then emulsify or immerse on the glass slide. After this, you have to let it air dry, means you have to just let it sit there for some time so that the smear dries out. It is no more wet. After that, the most important thing is you have to heat fix the slide. Now, how do we heat fix the slide? We heat fix either by passing this slide or 3 to 4 times through the flame of a burner. This is one method. Or if you are not using flame, you are using slide warmer, then just keep it on slide warmer for some time so that it gets heat fixed. Like in case of electric loop sterilizers, we don't use flame. There you can use a slide warmer to fix it. This step is important. Why it is important? Because if you don't heat fix the slide, then what will happen? Your smear will wash off. The bacteria will wash off while you are doing the staining. So heat fixing is important. Now once your slide is ready, it is heat fixed, you have to start the staining procedure. Let's see how we will do this gram staining procedure. If you are using fluid specimen, then since the concentration is very less, you need to centrifuge this specimen and use the sediment, that is whatever is settled at the bottom. You just use a drop of that bottom sediment and use it to make the smear. Now starting the procedure. First of all, you have made the smear. Now you cover this smear with the primary stain, which is most likely a crystal violet stain. So you pour crystal violet, which is purple colored, and let it there for 1 minute. After 1 minute, just wash it with plain water under running tap and then put your slide on the staining rack and cover it with grams iodine. You let this gram iodine there on the slide for 1 minute and again wash it with water. After that, we need to decolorize. This decolorization is the most important step in any staining procedure. Here what you will do, you will just pour the decolorizer, that is acetone, into the slide and let it wash away. After just pouring it, you will see that there is the primary stain will get washed away with it, and immediately you rinse it under the tap water. You don't have to put it and wait and let it sit there for more time. Otherwise what will happen? There will be over decolorization of the slide. If there is over decolorization, even the gram positive bacteria will lose the stain and it will appear pink in color. So you don't want the gram positive organisms to appear pink in color. You have to use this time limit very meticulously. Now once you have decolorized, washed it with water, then cover the smear with counter stain. Counter stain you have to cover for 1 minute. If you are using any commercial kit, then you have to look into the literature. They have a specific time frames, you have to follow them, otherwise it is 1 minute. Then again let it wash and let it air dry. So what is the principle for gram stain? It was first used by Hans Christian Gram, who was a scientist, in 1884. So basically gram stain is what? It is a differential staining technique. What it will do? It will differentiate, it will make bacteria differ on two aspects. So it will differentiate the bacteria either into gram positive or gram negative. So what happens that the crystal violet initially stain the bacteria purple. All bacteria will be stained purple, whatever it is. Then you use iodine. This iodine will form a complex with crystal violet. This complex will bind with the cell wall of the bacteria. Now what happens if there is gram positive bacteria? They have a different type of cell wall. There is a more of peptidoglycan layer and less lipid. So this crystal violet iodine complex will bind very strongly with the cell wall, and it will not wash away when you use a decolorizer. Decolorizer is an organic solvent. It will remove the lipids from the cell wall of the gram negative organism. Also peptidoglycan layer is very thin in case of gram negative organisms, so this will not bind firmly, it will not bind very strongly, and when we use a decolorizer it washes away this complex in case of gram negative, but it will not wash away the complex from gram positive organism cell wall. So when you take you put saffron in, what happens? If the organism is decolorized, it will take up the primary stain, saffron stain. If it was not decolorized, then it will keep it purple only. So this is the difference between cell wall of gram positive and gram negative. We have seen in previous videos. Peptidoglycan is 15 to 80 nanometer, that is it is thicker in case of gram positive, but it is only 2 to 3 nanometer, that is it is very thin in case of gram negative bacteria. Also lipid content is more in gram negative bacteria and less in case of gram positive bacteria. So this is again a picture of the two types of cell wall. On the left side there is gram positive, the blue colored part is peptidoglycan, which is very thick in gram positive. On the right side you see the peptidoglycan layer, that is blue colored, is thinner as compared to gram positive. So this we have already seen that gram negative bacilli will lose crystal violet, that is the primary stain, because of decolorizer, and that's why they will ultimately take up the secondary stain, that is the saffronin, and will appear. So what organism is this? What is the morphology of this organism? Here you can see there are purple colored circles, that is the cocci. Okay, this is not elongated, this is not bacilli, this is cocci form. So this is purple colored cocci, you call it as gram positive cocci. And what is the arrangement? We need to tell the morphology in terms of arrangement also. It is arranged in the form of clusters, like grape like clusters you see. So this is gram positive cocci in clusters, so we call it as Staphylococcus species. Here you can see there are gram positive cocci, but they are not arranged in the clusters, they are arranged in the form of long chains. So this is not a Staphylococcus, we call it as Streptococcus. So these are gram positive cocci in chains. Whenever there are gram positive cocci in chains, we think of Streptococcus species. Now here we can see there are gram positive bacilli. When we see gram positive bacilli, it could be Bacillus species or Clostridium species. If you can appreciate the spores at the end of the bacilli, then you can see these are not bulging spores, so most likely it is a Bacillus species. In case of Clostridium, the spores are usually bulging out of the cell. Now here we see pink colored rods. Pink colored rods means gram negative bacilli. This can be anything like E. coli, Klebsiella, Pseudomonas. So this is gram negative bacilli. Thank you. If you like the video, please subscribe to the channel and let me know in the comment section if you have any feedbacks. Thanks.